food and dairy microbiology. So microbiology, as you all know, is about the study of microorganisms. Microorganisms are very small in size that cannot be seen with our naked eyes. Only with the help of microscopes we can observe the microorganism. So the microorganisms are both beneficial as well as harmful. So the harmful microorganisms are the disease-causing microorganisms that are said to be the pathogens, whereas the useful microorganisms are the beneficial ones that are used for the production of many industrial products like dairy products, fermented dairy products like cheese, yogurt, and other industrial products like ethanol, all those things. Okay, so today I'll be explaining you about the different methods of food preservation. So before seeing about the preservation, certain terms should be known in food microbiology. That is, the first one is the contamination. Contamination is the presence of microorganism in the food. So the presence of microorganism cannot be identified in the food because there is no changes in the food takes place when the microorganisms are merely present in the food. So there are different sources of contamination of microorganisms in the food. So mainly it includes the raw materials. The raw materials includes the fruits, vegetables, whatever raw materials we use for the preparation of food. So the microorganisms may come from the raw material. The water which is used for the preparation of the food or else for the washing the fruits, vegetables, etc. If they have any waterborne organisms, that may enter into the food. And then from the air has many microorganisms that may also enter into the food when the food is kept open. And then sewage, rodents, insects, all these are the different sources of microorganisms in the food. Animals, birds, packaging materials, the materials which are used to pack the foods, as well as the handlers. So handlers are the main source. Suppose the handlers includes both the patients as well as the carriers who carry the microorganisms into the food. So the presence of microorganism in the food is nothing but the contamination. And then when the food gets contaminated, it may lead to spoilage. That is, the microorganism grow in the food. It utilizes the nutrients present in the food and it starts growing, multiplying, reproduce, and it changes the quality of the food. That is, the originality of the food gets altered. So that will be evident for us. That is, there may be change in aroma, change in taste, change in the appearance, etc. So this is when there is any changes in their originality, then it is said to be the spoilage. Okay. So the spoilage based on the that is the microbial spoilage, the time taken for microbial spoilage. The foods are classified into three types: stable or non-perishable foods, that is, that do not easily undergo spoilage; semi-perishable foods, that includes potatoes, apples; and the perishable foods that are easily spoiled like meat, fish, poultry, etc. The next one is about the preservation. So in order to avoid the microbial contamination and the spoilage, we have to preserve the food properly. So there are different methods of preservation. So preservation is nothing but protecting the food away from microorganisms. So there are different methods of food preservation. That includes asepsis, removal of microorganism; heat, that is high temperature of preservation; refrigeration or the low temperature of preservation; canning, drying, chemical preservatives, and radiation. So we will be seeing about each method. So first one is the asepsis. Asepsis is nothing but keeping the food away from microorganism. So that is, the microorganism will, that is, the food will be protected from the microorganism. Actually, by nature, the shells of nuts, skin of fruits, vegetables, membranes or fats on meat or fish, generally they protect the inner tissues of fruits and the meat. Okay, so this actually led to the discovery of packaging the foods. So when the foods are properly packed, the microorganism cannot easily enter inside the food. So this is the actually known as asepsis, keeping sterile, that is keeping the food away from microorganism. So this is one of the method of preservation. Next one is the removal of microorganism. Suppose if it is known that the food contains microorganism, that can be removed by different methods. That is, in case of if it is a liquid food like milk, beverages, etc., that can be, the microorganisms can be removed by either filtration, centrifugation, sedimentation, etc. So you might be knowing all these things, that is the what is sedimentation, centrifugation, like that. Whereas in case of if it is a solid foods like vegetables, fruits, etc., if any part is found to be spoiled, that can be trimmed. Okay. For example, trimming the outer part of cabbage, the spoiled layer of the cabbage, and then trimming the particular portion of apple. So all these are removing the microorganism from the food. So this is also one of the method of food preservation. Next one is by the use of high temperature or heat. So heat or high temperature, actually we all know the high temperature kills the microorganisms. Okay, it kills the microorganisms. Generally there are three different range of temperatures are used for killing the microorganism. The pasteurization is the one where it is a mild heat treatment process. That is, certain foods, it gets altered, the nature gets altered when it is subjected to high temperature. So those foods can be preserved by the pasteurization method. So this method was introduced by Louis Pasteur. So the name was given as pasteurization. So where a mild heat treatment, that is below 100 degrees Celsius, is used. So mostly this method of preservation will be carried out for preserving milk, beverages, etc. Okay. And then heating at about 100 degrees. So about 100 means reaching 100 degrees Celsius. Okay, so this can be attained by boiling the liquid foods, or else the roasting, baking, frying. These are the different methods where the temperature approaches 100 degrees Celsius. So when the food is approaching to the 100 degrees Celsius, the microorganisms present in the food will be destroyed. And the next method of preservation is the temperature where above 100 degrees Celsius will be used. That is, this above 100 degrees Celsius will be obtained only by the steam under pressure. That is the pressure cooking method, or else the hot retorts can be used, or autoclaving. So these are the different methods where the temperature above 100 degrees Celsius will be obtained, so that the microorganism, even the spore forming microorganisms that are not easily killed, can be destroyed. So the next one is the low temperature of food preservation. So the low temperature, actually they like high temperature, the low temperature of food preservation do not kill the microorganism rapidly, but in turn it slow down the enzymatic activity of the microorganism, so that the growth of the microorganism will be retarded. So there will be no growth of microorganism under low temperature, and once if it extends for a prolonged period, the microorganism will slowly will be killed. Okay. So this is about the low temperature. So in the low temperature of food preservation, different temperatures are used. So first one is the cellular storage, where about 15 degrees Celsius will be employed for preserving the food. So at this particular temperature, the foods like onion, potatoes, etc. can be preserved. Whereas the chilling or cold storage, where the temperature employed will be 0 to 5 degrees Celsius, that is the refrigeration temperature, where most of the foods are preserved, including the fruits, vegetables, dairy products, etc. And then the frozen or freezing storage, where the temperature will be less than 0, that is the minus degrees temperature, where the meat, fish, etc. can be stored. So this is about the preservation at low temperature of preservation. Then the next method is the drying method. So this is one of the oldest method of food preservation, where the food will be exposed to drying. So drying removes the moisture. So as you all know, the water is an essential one for all living cell. So likewise the microorganisms also require moisture for its survival. So once the water is removed from the food, the microorganism will be killed automatically because no moisture is available for its survival. So the drying method, this can be obtained. The drying can be done either by the natural method like solar drying or sun drying, or by the mechanical methods where the mechanical dryers are used. So mechanical dryers includes where the heat will be provided to the food, so which is a rapid method. So the natural method, the solar drying, takes time. It takes few days for drying. Whereas the mechanical drying, it gets completed within few hours because of the heat which is provided to the food. So the mechanical dryers includes spray drying, drum drying, foam mat drying, tower drying, etc. So these are the methods of drying. The next is the preservation by canning. So canning is actually preserving the foods in cans. So this method actually, in recent days, this has been become popular, the tinned foods or the canned foods. So this method was introduced by Nicholas Appert in 1809. So this is also, canning is also known as appertization. In the original method, they used bottles or jars, but this has been replaced in the recent years with tins and cans. Here actually there are different steps involved in canning process. So before doing the canning, in the beginning, cleaning of the food should be done. That is, in case if it is a fruit or vegetable to be canned, it has to be first washed to remove the dirt or any microorganisms present on the surface, and then it has to be subjected to blanching. Blanching is nothing but hot water washing. So when hot water is used for washing, the microorganisms gets eliminated, and finally the cans are filled with the food substances to be preserved, and after filling the cans are closed and sealed, and finally a heat treatment will be provided to the cans so that if any microorganisms are left, will be also destroyed. So here actually in the canning process, a vacuum will be created inside. Vacuum is no air will be there. That is, oxygen will be completely removed, and sometimes it can be replaced with other gases, hydrogen gases, etc. So that there will be no air for the microorganisms to grow. So the aerobic microorganisms cannot grow in the canned foods. And the next method of food preservation is the chemical preservatives. So you all know the chemical preservatives are added to the food to preserve the food. So these chemicals actually it inhibits the growth of microorganisms. So there are two classes of chemical preservatives, class 1 preservative and class 2. So class 1 preservatives includes the simpler ones like sugar, salt, spices, vinegar, honey, and vegetable oils. Whereas the class 2 preservatives includes organic acids like benzoic acid, as well as the salts of organic acids like sodium and potassium, salts, sorbic acid, and sodium and potassium, salts of sorbic acid like sorbates and benzoates. So these are the chemical preservatives. So when these chemical preservatives are added to the food, it preserves the food away from microorganisms. And finally, the last method of food preservation is the irradiation, that is the radiations. So this method of food preservation is also known as the cold sterilization method because no heat is produced there. So it is also a cold sterilization method. So it is also somewhat related to the pasteurization method because of the mild treatment. So the radiations which are commonly used for the preservative methods are gamma rays, X-rays, etc. So they actually inhibit the microorganisms or destroy the microorganisms so that the food can be preserved away from microorganisms. So today actually I have explained about the different methods of food preservation. So hope you all understood. Thank you for this opportunity.